Good morning, students. Welcome to the biology class 12. We are dealing with chapter 9, that is strategies for enhancement and food production, and this is part 3. So we have finished with part 1 and part 2, where we had dealt with the animal breeding. Okay, so today we are going to deal with the plant breeding techniques. Okay, and you should know what is the need of the plant breeding and why India, a country like India, needs to have more stress on the plant breeding techniques. So in this class, we are going to deal with what are the different steps through which we have to go through while doing the process of the plant breeding. Okay, have a nice day, everyone, and stay home, stay safe, so that we can have a good future. Okay, so let's come to this topic, today's topic, that is about the plant breeding. Now, what is the definition for plant breeding? The plant breeding is the technique that we are using to manipulate a plant species so that we can create a desired plant type. I think it's clear for you. What do you mean by the word manipulate? We are using our technologies, we are using our techniques so that we can create a plant species that contain the desirable qualities of that plant. And why you want to do like that? We are doing like this for getting a better yield and the disease resistant varieties. So can anyone tell me what is the main issue that we are facing while dealing with these plants? If you have a plant, if you have a garden in your home, you can see that after some days some of the diseases are affecting the plants and we are not able to cope up with. So what we do, we apply insecticide, we apply pesticide to kill these insects. So the best thing, yeah, the first thing that has to be done in the plant breeding is that it should be disease resistant. Okay, and of course we have to aim at the yield, the production, and the disease resistant varieties. And even one more thing you can say, the tolerance to the physical activities, like if the temperature has been increased, if the temperature has been decreased, the plant has to resist to these conditions. Okay, so for these purposes we are doing the plant breeding technique. So we are manipulating the plant species so that we will get the desired qualities in the plant. I think it's clear what is plant breeding. Now, what are the advantages of the plant breeding? Let's see one by one. What are the advantages? First one, to increase the crop yield, to improve the quality, to increase the tolerance level that we already discussed now. What are the different environmental issues? Salinity, then the extreme temperature, and the drought. Drought means the lack of water. Okay, and salinity means sometimes the salt content in the soil will increase, that will lead to the destruction of that plant. Okay, so these things we have to take care, and to resist to the pathogens, especially the attack by a virus, the attack by a fungi, and the attack by the bacteria. Then, the last not the least, to increase the tolerance to insect pests. Okay, if you have an insect pest, you have to increase their tolerance level. So these are the advantages. Yeah, you can say these are the aims that we have to keep in our mind when we are doing the plant breeding technique. So once again, we can say what are the advantages of the plant breeding? To increase the crop yield, to improve the quality, to increase the tolerance to different environmental stresses like salinity, temperature, and the drought, and to resist to the pathogenic attack like viruses, fungi, and the bacteria, and to increase the tolerance to the insect pest. Okay, you can write in your copy what are the advantages, yeah, what is the aim of doing the plant breeding techniques. So now we are going into the plant breeding technology. So these are the steps you have to follow. Each and every steps is important in the process of plant breeding. So what are the different steps? The first thing that we have to do is the collection of genetic variability. Okay, we will discuss each and every topic in detail. So now just see collection of genetic variability. And the second one, after getting that variability matlab the whole group of biodiversity, we will evaluate and we have to select the parents in from that genetic variability. What what is your aim? Yeah, what quality you have to be in that parent? You have to select that parent only. Okay, now after selection of the parent, the next step we have to follow is cross hybridization between the selected plants. So whatever plants we have selected, now the second step, third step is we have to hybridize. We have to do the hybridization between these selected plants. And once the hybridization has been done, the progeny has been obtained. Now, what is the next step? The selection. So, out of these progeny, we have to select, and once it has been selected, again it has to undergo a testing so that we will be easy to understand which is the superior recombinants. Previously, we have done with inbreeding. So, in inbreeding, we have said that it has to be breed for continuously 4 to 6 generations so that finally you are getting a pure line. Okay, the same process we are doing here. And what is the final step that we have to follow here? That is the testing. Once again, you have to test, then you have to release, and finally you have to commercialize the new breed of your plant into the market. So that is the final step. So before commercializing, it's very important to go through other steps. If any fault, any flaws happen in any of this step, then it will not be able to, will not be able to do the commercialization process. Okay, so these are the steps, and we will deal with each and every steps in detail. So first step that we are going to deal is collection of genetic variability. So genetic variability we normally take from a wild varieties. Yeah, they are wild relatives of the crop. You have to go to a germplasm bank where you can see a diverse collection of the seeds or diverse collection of the genes that can give the desired quality in that crop. So first thing that we have to do is that we have to go to a germplasm bank, we have to go into a seed bank, and you have to check what are the different variabilities that is available there. And normally we take the variabilities from the wild varieties. So these wild varieties and all the germs, all the alleles have been preserved in the germplasm bank. So we are visiting that germplasm bank and we are collecting the desired qualities, and that characters have been evaluated. So let's see what are the different steps. Genetic variability normally we get from the wild relatives of the crops. Okay, now all the wild relatives, wild varieties, we are collecting them and we are preserving them. Their characters are evaluated, and this entire collection from which we are getting all these characters are called as the germplasm collection. Okay, so that is the first step that we have to undergo, that is a collection of genetic variability. So this is a germplasm bank. I'm showing these pictures so that it will be easy for you to identify what do you mean by a germ. You might have heard about the blood bank and all, a sperm bank, blood bank. Now this is the germplasm bank where we are preserving the genes, alleles of the seeds, any of the plants, so that it can be used for the later, future purposes. Okay, so this is a seed bank, this is a germplasm bank where we can see a wide variety, and it has been labeled properly because this label will tell you what are the different characteristics that seeds contain. Okay, and out of these one, we have to select the superior qualities, the qualities that you want to be in that plant, that will be selected, and we have to evaluate that. Okay, I think it's clear for you. The first point, that is a collection of the variability. Come to the second point, that is evaluation of the selection of the parents. So germplasm has been evaluated and we are evaluating what are the characteristics, what are the desired characteristics that we want to have in that plant. Okay, and this plants that we have, so you have to raise that plant, and this selected plant, they have been multiplied and they have been used in the hybridization. How we can multiply? It's not an easy process. Okay, they are writing only one word, that is multiplied, but how you can multiply it? You have to undergo the inbreeding techniques. Okay, so what is inbreeding? You are crossing them between the same breed, and finally after crossing for 1 to 2 generation, 2 to 3 generation, you are getting the pure line, and this pure line will contain all the desirable qualities that we are expecting. Because why we are doing inbreeding? If any other undesirable characters are there, it can be removed after undergoing a successful inbreeding. Okay, and again, it should not undergo into an inbreeding depression. Up to the 5 generation, we can do the inbreeding so that finally we are getting a pure line. And this pure line is created where it's contained all the desirable alleles. So here only one word has been given, that is multiplied. And how we can multiply? By repeated inbreeding. Okay, So the germplasm has been evaluated. We are selecting the combinations of the characters, and these selector plants we are multiplying and by the method of inbreeding, and finally after inbreeding we are getting the pure lines. Clear? I think it's clear for you. Just connect this evaluation of the seeds along with the inbreeding. Then only we will get the pure lines. So I have written here what are the characteristics of the pure lines. So all the plants within a pure line, they have the same genotype as from which plant we have derived from. Okay. And this is because the parent plant was homozygous and they are undergoing the self-fertilization process. You can note down in your copy, yeah, what is a pure line? Yeah, what are the characteristics of the pure line? So after doing inbreeding, after doing self-fertilization, we are getting the plant in which all the desired qualities have been incorporated. Okay, and that is called as a pure line. So after selecting this one, we are able to raise the pure line. And this pure line will go for the further process, that is for the process of the hybridization. Okay, so the most important thing is that we have to get the pure line. Without the pure line, we cannot proceed further in the plant breeding techniques. So the next one, the third point, yeah, the third step that we have to face is the cross hybridization among the selected parents. So already the parents are ready with the pure lines with the desired characters. Now we have to do the hybridization process. What is hybridization? The crossing between the superior quality parents. Okay, so the selected parents are cross hybridized. And what method normally we use? We use the technique of emasculation and bagging. Okay, and the hybrid that has been produced after the cross hybridization, they combine the characters of both the parents. But one more thing, it's a disadvantage or drawback. Don't think that every step is going to be a successful step. It's all trial methods. Okay, trial and error methods. So once you have failed in that step, you have to go back to the first step. Again, you have to get into the germplasm, you have to get into the inbreeding, you have to raise the pure line. So it's a little bit time taking process. Okay, so the hybrid that has been produced, it should contain both the characters. Otherwise, it is of no use. Why we want to undergo all these processes? So while doing the cross hybridization, emasculation method, what is emasculation method? You are selecting two parents. Okay, and one parent you want their pollen grain and other parent is good in their developmental stages. So you are collecting a desired pollen grain and you have to dust that pollen grain on the stigma of the another plant of the same species. Okay, and while doing that, if it is a bisexual flower, you know that both the sexes are present, they have their own pollen grain. You have to remove the self pollen grain first. If the self pollen grain falls on the flower, then you are not going to get any hybrid variety. So first thing that we have to do is that if it is a bisexual flower, you have to remove the pollen grain of that flower. And then you are selecting a pollen grain of the hybrid variety, your high quality, and you are dusting that pollen grain into the stigma of the another flower, and this process is called as the emasculation. So you are taking out the anther and you are taking out the pollen grain and you are dusting that pollen grain on the stigma of the flower where the development has been expected, and that process is called as emasculation. And after doing the emasculation, you have to cover that pollinated one, that pollinated flower with a bag, so that any other foreign pollen grains will not fall on it, and this process is called as the bagging. I think it's clear for you. So the first thing that we have to do is emasculation. After doing the emasculation, we have to do the process of the bagging, where we are covering the plants properly with a bag. It can be a polythene cover. The only aim is that to stop other pollen grains from contaminating that flower. Okay. So see this picture, I'm showing this picture so that it will be clear for you. This person is having a brush, and what he is actually doing with the brush? He is collecting the pollen grain, and this pollen grain is going to dust on the another flower. Okay, and this process is called as the emasculation. And after emasculation, this flower will be covered with a polythene cover, yeah, any type of bag, so that another pollen grains will not fall over it and contaminate that flower. This process is called as emasculation. You can try at your home also. If you have lots of flowers in your garden, you can try with this method. If you have a proper brush, you can collect the pollen grain of a flower. It has to be of the same species, and you can dust it on the another flower so that the pollination can take place artificially. And these are all artificial method. We are not waiting for any pollinating agents. It's going in a artificial method. Clear? So this is called as the cross hybridization. So this way we are doing the hybridization. And again I am telling, at the end of the hybridization, don't expect that all the crosses that we have made is going to be a successful one. Many of them will be a failure, only very few will be having our desired product. So I've given here what are the advantages and disadvantages of cross hybridization. You can write this in your copy. So what are the advantages? In many crops, the F1 hybrids are early in maturity. They produce goods with uniform size. They are resistant to biotic and abiotic stresses. They are always the high yielding varieties. Now, what are the disadvantages? The production is high. The fresh seeds that we are taking, we have to purchase, we have to buy at every time. Sometimes the F1 hybrids are vulnerable to the diseases. So these are the advantages and the disadvantages of the cross hybridization. Now, the fourth step is we are going to have one more step, that is a selection. And after selecting, we have to again test for the superior recombinants. So again, one more selection has been, and so once we have cross hybridized, we got the product. That product again we have to select among them by doing one more cross hybridization. You have to be very careful in evaluating the desired qualities. And what is our expectation? The hybrid plants that is going to arise from it, they should contain the superior qualities of both the parents. Okay. So once the cross hybridization has done, you have to select it, and the selected progeny has to undergo another round of cross hybridization. Clear? So see, selection we have done once the cross hybridization. Here I have given a whole set of the seeds. They have to select from this. You have to select the superior qualities, and again you have to do cross hybridization. So selection process is crucial to the success of the breeding objective, and they are self-pollinated for several generations so that they will reach a uniform stage, a pure line stage, where they will not undergo any kind of segregation. Normally, you didn't study about the Mendel's law, but when you study this Mendel's law, we will see that the characters should not segregate. If it is getting segregated, if it is right, if it is getting united, then the desirable characters will not be there. So the characters will not segregate in the progeny. And the last step, so cross hybridization, then selection, again cross hybridization. Finally, we are going to test and release and the commercialization of the new cultivars. So the selected hybrids have been evaluated for the yield, quality, and disease resistance. And this evaluation you can do in any of the research fields and record their performance. And testing is done in the farmers fields, because up to this time we were testing in a lab. We are using only the lab methodology. But after this, what you have to do, you have to transfer these plants into a soil, that is a real place where a farmer can grow the seed. So we have to transfer this one into the soil. So that's why it is called as farmers field, and you have to test it for at least for 2 to 3 generations. Again, it's a very time taking process, because once it has been released, it should not be have any of the disadvantages. So you have to do at least 3 generations of the testing. And finally, after the 3rd generation, the seeds that we are getting, that can be called as hybrid seeds, and they are marketed to the farmers. Clear? It's farmers, it's farmers. Okay. So that is a final step, testing, release, and commercialize. Now, what you understand from seeing all these steps are all time taking. We should say these are all time taking, because as we have been given the steps, it's not easy to have each and every step, because if once we failed in any of the steps, we have to go back to the first step again. Okay, and it's a mainly out of the 100 plans we are trying to raise, only 10, yeah, 15% will be a successful one. So you have, because it's a segregation of the characters, and many Mendelian characters can come inside that. So it's a time-taking process. Clear? So you have to go through all these steps, and finally, these are the steps of the plant breeding. Okay, this is what the evaluation, this is called as a farmer's field. I have shown the picture here, the farmer's field. So whatever we have done, like preliminary breeding, then evaluation and selection, now it's in the hands of the farmers, because the farmers are the one who is going to develop these seeds. So farmers participatory selection. You can display the seeds that you have developed, and from there, the farmers can select that, and finally the commercialization. After commercialization, you are developing the plant. And this is the farmer's place where we are going to have a trial, and we are seeing how the plants are going. And this has to be done at least 3 generations. You can see there are a whole lot of plants here, all are looking different. So that means they are having testing. It's not commercialized one, they are doing the testing process. You can see different plots, and in each plot they have growing that seeds to see what is the result. Okay, so that's all about the plant breeding techniques. So you have to learn all the steps of the plant breeding. Okay, the steps are not difficult, but if you are doing any plant breeding in a real life, that is difficult. But the steps you have to learn. Okay. Next one is green revolution. I think you are learning from the smaller classes what is green revolution. It's related to the green plants, especially the wheat, maize, and the rice. Okay, so it's mainly dealing with the wheat and rice. The development of high-yielding varieties of wheat and rice in 1960, when we have faced a lot of scarcity for the food material. And what is the aim of this green revolution? To increase the food production. And the word green has been given because it's all plants, it all dealing with the wheat and the rice. So that is coming under the green revolution. Now, wheat and rice, we can see what are the different varieties that we are growing here. Now, tell me what variety, yeah, how a wheat and rice plant looks. Yeah, what is the first quality that we are expecting? Of course, the quality like protein, carbohydrate, everything should be there. But the one important thing is that the plant should not grow too tall. Yeah, the plant should not be so short. So what is the best option we are having? We can create the plants having a semi-dwarf varieties. If it is too tall, what is the disadvantage? If a wind blows, it can fall down easily. And if it is too short, your rodents, rats, mice, any of these people can attack, and it can eat all the grains that we are having. So what is our best option? What we are trying to do, we are trying to have a semi-dwarf variety. Okay. So increase in the wheat and rice production by using the semi-dwarf varieties. And the semi-dwarf varieties was developed by Norman E. Borlaug in the Mexico. And we have the two high yielding varieties, Indian varieties that we are using here is Sonalika and Kalyan Sona. You have to learn this name. Okay. And about the rice also, they have developed the semi-dwarf varieties of the rice, IR8 and the Taiching Native 1. And they developed in India. The semi-dwarf rice varieties that develop in India is Jaya and the Ratna. So Indian varieties of wheat is, which are the two Indian varieties of wheat, Sonalika and Kalyan Sona, and which are the two varieties of the rice in India, that is Jaya and the Ratna. Okay, and what is the important quality? They have the semi-dwarf varieties. And the next one is sugarcane. Here we are dealing with two types of sugarcane. One is the Saccharum barberi. Okay, their scientific name is Saccharum barberi, and other one is the Saccharum officinarum. Always this question used to come for your exam, like what is the difference between the barberi and officinarum. Okay, so let's see. So Saccharum barberi, it's actually, it will grow in North India, and their qualities, we can say they have a poor sugar content and the yield. Okay, they have the poor sugar content and the yield. And what about the Saccharum officinarum? It is growing in South India. Okay, so this is North India, and here it is South India, and here it is having a poor sugar content, and here it is having a higher sugar content. It's poor sugar content, it's having higher sugar content. But the drawback is that they will not be able to grow in the climate of the North India, because South Indian climate is different from North India. So this Saccharum barberi cannot, sorry, Saccharum officinarum cannot grow in the North India. So what is our option? We have to cross between these two breeds. So we have crossed these two breeds, and what we got now, they had a thick stem, because if it is a thick stem, your content of sugar will be more, and high sugar content, and your high yield and ability to grow in the North India. Okay, so there are two species, two different species. One is Saccharum barberi and other one is Saccharum officinarum. So Saccharum barberi is growing in the North India, but their quality and their sugar content is very less. But the Saccharum officinarum, they are growing in the South India. They have a very thick stem and their sugar content is high. So the best solution we are having is that we are crossing these two species and we are developing a hybrid which contain all these qualities, and that can be grown in the North India also. You can see the picture also. The stems are looking thick, and of course it might contain the more sugar content, and it can be easily grown in the North India. Okay, so that's all about this plant breeding techniques and the varieties of wheat, rice, and even the sugarcane. Now let's see a video in which all these things have been described. Okay, like what are the 5 steps that we have described in plant breeding? The first step, the collection of germplasm. Second one, selection of superior parents. Third one, cross hybridization. Fourth one, selection of superior hybrids. And the fifth one, evaluation of the selected hybrids. So let's see this video. And if you want, you note down the points. It's already been discussed earlier, just a replication of that, so that you can visualize that is in front of you.
First step in plant breeding is the collection of germplasm of a particular crop that is intended for breeding. Germplasm is the collection of all the genes of different varieties of a particular crop in the form of seeds or plants. In this way, the naturally available gene pool can be utilized for breeding. The second step is the selection of superior parents. Here, 2 plants with the different desired characters are selected from the available germplasm. Then, they are made to self-cross to get a pure line of those varieties. The third step is cross-hybridization of the superior parents. This is done by collecting the pollen grains of one parent plant and placing them on the stigma of the other parent plant. However, the hybrids obtained may not contain all the desired characters present in the superior parents. In fact, 1 in 1,000 or 2,000 hybrids obtained may contain the desired characters of both superior parents. Hence, this step is considered cumbersome. The fourth step is the selection of superior hybrids. The desired hybrid is selected from the thousands of hybrids formed during crossbreeding that possess the characters of both its parents. Hence, it is considered superior than its parents. The hybrid is then allowed to self-pollinate till it forms a pure line of homozygous plants. The final step is the evaluation of the hybrid for the characters gained. This is done by first cultivating the hybrid in a research field under ideal conditions. Later, they are cultivated in farmers' fields at different locations for 3 growing seasons. This helps in comparing the performance of the hybrid against the best crop cultivar available in that area. In this way, plant breeding has increased the yield of many crops, including wheat, rice, sugarcane, and millet.
I think the video is very clear for you. All the 5 steps have been showing here with the practical knowledge of how they are doing this process of the plant breeding. Okay, and please keep this video in your mind while studying, so it will be easy for you. What do you mean by 3 rounds of the selection, and also it will be in front of you. The farmers are raising that crop for 3 times, then only finally they are commercialized. I think all these topics are clear for you. If any doubts, you can ask me. And even though it's look like a symbol, the procedure, if you are doing for the correct one, it will be difficult in the experimental level. But for learning purpose, it is good. And you have to go through each and every topic, and the each and every scientific words you have to use while writing this one. It can come for an essay also, like what are the different plant breeding technologies. Okay, so that's all for today's lesson. Go through this plant breeding technology along with the video. And do well, everyone. If any doubts, you can clear me later. Thank you, students.